let's talk about Mendelian genetics. This is the type of genetics you've probably learned about before. It talks about things like alleles that are dominant versus recessive and that sort of stuff. So we call it Mendelian genetics because the earliest understanding of genetics came about because of research by the monk Gregor Mendel. Now, Gregor Mendel worked with peas, garden peas, which genetically speaking are relatively simple organisms. And by crossbreeding and keeping insanely meticulous records, he was able to figure out these two important laws: the law of segregation and the law of independent assortment. So the law of segregation means that offspring get one copy of a gene, one allele from each parent, one from mom, one from dad. We now know this is because our genes, we have two copies of them and they're on chromosomes, and you get one chromosome from your mother, one copy from your father, each with one gene, which can have a lot of different alleles or variations. The law of independent assortment states that which copy of their parent's gene an offspring gets is random. The alleles from a parent are passed on independent of the other alleles that are inherited. So if you have an offspring, so you have a child, if you give that child the copy of chromosome 1 you inherited from your father, that has no impact on whether you give that child the copy of chromosome 2 that you got from your father or not. Which copy of the chromosome you pass on is a completely random process. This means, believe it or not, you may not have any genetic material from your great-great-great-great-grandmother. Genetic ancestry and cultural ancestry are two different things. All of these DNA kits that promise to tell you where your ancestors are from are ignoring the fact that your genes might not necessarily reflect that. And the other problem with the law of independent assortment is when you start getting to complex organisms like humans, we have multiple genes on our chromosomes. And if two genes are close together on the same chromosome, they tend to go together. So you may see alleles that tend to coexist. Blonde hair and blue eyes, for example. Those tend to go together. They're probably close together on the same chromosome. Okay, I like numbers. I'm a total nerd. I know it. I admit it. When we look at the law of segregation and the law of independent assortment, we can do math that says that each person can produce 8,388,608 unique gametes. Gametes are the sperm and the egg. So that's a huge number. 8 million unique sperm. Women generally don't produce this many eggs, but that strongly suggests that each of those eggs is genetically unique. Now I've got a note here that says not considering the effects of crossing over. We're going to get to crossing over, but it's something that happens during meiosis, or the production of sperm and eggs, that increases the diversity of our gametes beyond 8 million. Wow. So if a man can produce over 8 million unique gametes, a woman can produce over 8 million unique gametes. They get together and have an offspring. Look at the size of that number. Wow, let's see. There are 70 trillion different possible offspring. Whoa! And again, this is not considering the effects of crossing over, which we're going to get to in just the next slide. This number is significantly larger than the estimated number of how many humans who have ever lived. Wow. So everybody who's ever lived has been genetically unique. Okay, so now for crossing over. During meiosis, remember, meiosis is the process where cells go from being diploid, or having 2 copies of each chromosome, to becoming gametes, sperm and egg, that have 1 copy of each chromosome. We call that haploid. During this process, cells duplicate all of their chromosomes, and they use cell division to reduce the number of chromosomes. Okay. But what can happen is that 2 chromosomes, the 2 different copies of each chromosome, during meiosis, they can swap part of their chromosomes. So if we look at chromosome 8 here, you see this big red area? That indicates that at some point, one of my ancestors, chromosome number 8, crossed over with the other copy of chromosome 8 in that individual and made this new genetically unique chromosome 8. Yes, these are my chromosomes. And you can see largely Northwestern European, good chunk from some of the Southern European areas. This red part, Sub-Saharan Africa, West Africa. I can tell you this means that at least one of my ancestors was probably brought over to the US through slavery, which means that some of my other ancestors were probably slave owners. In fact, that's pretty likely given how long they've been in the country. Now, if I had kids and all of my kids got this all blue copy of chromosome 8, they would not have the genetic heritage that shows that ancestry. So just because you don't have genes from a certain area doesn't mean you don't have ancestors from there. Again, cultural and genetic heritage are 2 different things. So anyway, this crossing over process really increases the genetic diversity even further than just the laws of independent assortment and segregation. So the Mendelian understanding of genetics. I've written this on here. I've put these little lightning bolts. It doesn't apply to most human traits. This work was done in peas. It's very, very important because it helped us understand some of the basics about genetics. But we've gotten well past that. And when we look at human traits, we see a lot of more complex patterns of inheritance. So in Mendel's P's, for some of the genes, one allele will be what we call dominant, and another allele will be what we call recessive. Dominant recessive is pretty easy to understand. If you have one copy of the dominant allele, you're going to have that characteristic. If you have two copies of the dominant allele, you will have that characteristic. For the recessive characteristic, you will only see that. You will only have that characteristic if both copies of the gene are recessive. When we talk about these combinations, we have some fancy words. If your alleles, your two copies of the gene are different, we call that heterozygous. Anytime you see hetero in science, it means different. If you have two copies that have the same allele, we call that homozygous. Homo in science means the same. And recessive alleles can be alleles that are not functional for some reason or another. They don't produce a functional protein. And so you don't see that lack of functional protein unless you just have that recessive allele. Now, in humans, most traits have multiple genes controlling them. Look at all the red stars I put on this slide. This is an important slide. Eye color, there are around 16 genes that we know of that determine eye color. If it was just brown versus blue, everybody who had blue eyes would have the same color blue eyes. But we don't. We have a lot more diversity because there are so many genes involved. Two of these contribute the most, but the rest influence the variation. So the blue versus brown is the biggest component, but dark blue versus blue-green versus light blue, that's all the other genes involved. Most genes have more than two alleles. So it's not just a dominant allele and a recessive allele. The cystic fibrosis gene, for example, has over 1,000 known alleles. So that's 1,000 different versions of that gene, some of which don't cause cystic fibrosis, others of which do. And if you have problems with the cystic fibrosis gene, how severe the illness is depends on which alleles you inherited. Most importantly, dominant recessive is the exception to the rule in most organisms. Even in most plants, the P's are actually exceptional for how simple their genes are. Okay, so that brings us to the end of the non-Mendelian genetics. I want you to remember the information on this slide because genetics, it's more complicated than that.